So hello and welcome again. In this video I would like to talk about the reproduction in flowering plants. So plant reproduction. And there are different types of plants, of course, but I'm now not going to refer to the mosses or the ferns and also not to the conifers. I'm referring to the so-called the flowering plants or angiosperms. Okay, plants, and these are also known as angiosperms. Okay, if you remember the option, there are also mosses and ferns and conifers, yeah. So what I would like to do first is I would like to talk about the structure of a flower. So flower structure, after all, it is the flower which is the reproductive structure of a plant. So, excuse me. I'm going to make a drawing of a very schematic drawing of a flower, cross section. We'll start like this. Okay, whoop. Okay, so that is the central part of the flower. This is known as the style and the stigma. I'm going to label this later. And over here, I'm going to draw schematically. These are called the so-called the petals. A little bit distorted. Okay, they are usually colorful. And over here, of course, like this. Okay, that's yeah, the stem. And what I also have is I have a bunch of anthers as well. So this is basically my drawing of a cut open flower, and what I'm going to do now is I'm going to start labeling the whole thing, and we're going to start here at the top. This top part here, this is well, first of all, this central part here in the middle, okay, that is the female part of the flower, okay, and this top part here, this is where the pollen lands, and this has a name, it's called the stigma. And this long central part here, that is known as the style. And both of these together, it's the female part. Okay, that's the sign for female. And what we also have here is these here. Yeah, that's the anther. Okay, it's an anther, and that's the male part. So those anthers, they produce pollen. And these are known as the petals, and usually they are quite colorful, okay, so to attract insects. And something that I did not mention here, down here there are also leaves. They are green and they have a name, and they're called sepals. And the question is now, where are now the ovaries? The ovaries, this is the ovary here, this large part here, this is known as the ovary. And inside the ovaries, those dots here, these are the egg cells, they are known as ovules. Okay, and with that, I would like to explain to you a little bit. You have to know the structure of the flower here. I would like to explain to you a little bit of what actually happens here. When you see a flower, and you have pollen grain, usually from another flower, and then these pollen grains, okay, let's say this is a pollen, and these pollen grains come from the anther, usually of another plant. Okay, and these pollen grains, they will land here on the stigma, and the stigma is sticky. And what's going to happen is that those pollen grains, they will form a tube. There are tubes growing all the way down the style, and in this tube you will have the sperm cells traveling. So it's like the pollen will start. I've got here pollen. Okay, and so if this is a pollen, let's say under the microscope, what's going to happen is that actually a tube will start to grow. Okay, slowly. So, and this tube goes all the way down, and in this tube you have then the sperm cells. Okay, So the pollen will start to grow a tube down the style, and in this tube you have the sperm cells, and then what you have is down here in the ovules you have fertilization taking place. And what will happen later on is that these ovules, after fertilization, they will form the seeds, and the ovary later on will form the fruit. Yeah. So basically what you have here is you have a difference between pollination, which is the transfer of pollen from one flower to another flower, and you have down here in the ovary at the ovules you have fertilization. So fertilization happens here. Okay, this which happens down here, and up here you have pollination. Okay, so this actually shows that pollination does not always lead to fertilization. So there are already two terms that I have covered here. Then what you have forming is you have a fruit forming. And in case you're just wondering, next time when you eat an apple, what you're going to see is, of course, this is, I'm going to draw an apple now. Oh my gosh. Okay, if this is supposed to be the apple, okay, then you will actually see down here those little, tiny little leaves on the bottom side. Yeah, and those leaves actually, these are, the leftover of the sepals. And this one here, that was the apple itself. This once was an ovary, and the seeds inside the apple, these were actually once the ovules. Yeah. And that's of course the stem, so it's actually you can see over here that the ovary actually started to grow bigger and bigger and bigger and bigger. Okay, what will happen then is that you will ultimately then have a fruit. Okay, and this actually leads us to the next thing, and which is known as seed dispersal. How do you actually spread the seed? And for seed dispersal, you have the fruit. The fruit is very important for seed dispersal. Now, the question is now, how are the seeds of an apple dispersed? The apple falls to the ground, and then an animal eats the apple, and this is how the seed is dispersed. So seed dispersal can, of course, be done by animals. The seeds can be dispersed by wind. Okay, you know these things, huh? Look at this. Does this look familiar? Something like this? Yeah, from a dandelion. Okay. It's a wind dispersed. You've got the seed here. What do you think this is? That's the fruit. Or for example, you've already seen those, it looks like this, and you've got the seed here. Yeah, and this falls from the tree and it starts to rotate around in the spiral, right? Well, that here is the fruit. And it's the seed, of course. Yeah. So the thing that I'm trying to clarify here is that the word fruit, you have to kind of leave this a little bit, the old idea of what a fruit is. Like when we say fruit, we think, you can eat it. Well, that's only one possibility here. There are different ways of, fruit, but from a botanical perspective, is the structure that forms out of the ovary. Okay? So this is kind of the idea here is we've talked about three things now. Okay, we've talked about pollination. Should I write this down again? Pollination. We talked about fertilization. And we talked about seed dispersal. Okay, seed dispersal can be done, of course, by wind, by water, by animals. Okay, you think of it. What about pollination? Pollination can be done by wind. It can also be done by animals, like insects. Yeah. So this is should kind of illustrate to you a little bit that these are three different concepts here, and that pollination does not automatically lead to fertilization, and of course, not every fertilized ovary will form a seed to be dispersed. Okay, so this is simply something that I just wanted to clarify here. Okay, so this is basically wraps up already two concepts that I wanted to talk about. And the next thing that I would like to talk about is something called mutualism. In pollination. In very simple words, this basically means that insects and flowers they have to sometimes work together for pollination to happen. There's a mutual benefit. The insects benefit from the flower, and the flower benefits from the insect. So it's also known as symbiosis. And basically, this means that if an insect dies, then the plant dies out. It becomes extinct, and the plant will not have the possibility of being pollinated, because no other insect will be able to fit into the plant, for example. Okay, so this means that around 85% of flowering plants are insect pollinated. So this, the consequences are pretty intense. You spray poison to kill off the insects, you're killing off the plants, because they cannot the pollination, plant reproduction cannot happen. As a matter of fact, just in case you're wondering, this is the reason why, for example, people who are growing fruit trees and so on, they sometimes keep beehives next to it, so that you're able to, so that they will actually do the pollination. Yeah? And I also heard in sometimes when they don't have a beehive, and because there are no bees, because of, I don't know, maybe it's too cold or there's something else was wrong, then what some farmers have to do is they have to manually pollinate the flowers. So what they will do is they will take a small brush, they will actually pick up some of the pollen here, so they will take, this is supposed to be a brush, okay, there are specific brushes. They will actually use a brush to pick up the pollen here, and to transfer this pollen to the stigma of another flower. So they will do this manually, because the bees are not around to do that. Because if they don't do that, they won't have any fruits later on. So I just want to make this clear, that this whole issue with insects and being flower pollinated and so on is pretty important. Okay, so we talked about the next thing. Let's talk about a seed. So that's the next topic, is seed structure. And we just think of, I don't know, a bean maybe, or peanuts. This is actually quite an interesting thing. You won't be able to eat peanuts anymore the same way. But I'm going to just start here. I'm just going to draw a bean here. But actually it's the same for peanut. Okay. And what you have over here, this is the place. There's this, usually this part is a little bit whitish here. And this is, as we know, this is called the scar. It's like the place where this seed was attached to the ovary. Okay, seed attached to ovary. Okay, so that is the place. The seed is mostly actually made of starch. Okay, on the outside here, the next time when you eat beans, you will actually be able to see this. The outside there is known as the so-called the seed coat, and this has a name. It's called testa. It's the seed coat. Yes, think of peanuts. Okay, this is brownish, thin paper-like thing that's around on a peanut. Right, that is the seed coat. And then what we're going to do now is we're going to now look inside the seed, and I'm going to draw the seed again here. It's a little bit not so nice here. Okay, let's open it up, and what we have inside here is a little bit not so easily visible. It's maybe a little bit too large, but what you have is you have already here the so-called the embryo. It's like a mini plant. It's known as the embryo. And the embryo has a small shoot. Okay, and this has the name. It's known as plumule. It's like a tiny shoot, and this one down here, it's the root. Okay, and this root is known as the radicle. the radical. Okay, and but you see that the largest part here, of in this case of the bean, is a storage structure, and this will actually form the first two leaves. It's known as the cotyledon. In German, you call it Keimblätter. Yeah, and there are, in this case, there are two of them. Yeah? And again, think a little bit of your peanut. If you have a peanut, I'm going to draw, this is a bad peanut. Yeah? You draw a peanut, and there's this little thing here when you break it apart. Yeah? And this is actually the embryo. Yeah, so when you first you remove the tester, the brown paper-like seed coat, and then you have two halves of the peanut. And when you break it apart, there's this round little thingy there, and it's the embryo. So every time when you're eating peanuts, you're eating embryos. Okay, so just to clarify this a little bit. And of course, a seed that looks like this is known as has two cotyledons. It's known as a dicotyledonous plant. It's known as a dicot. Okay, and what will happen is if you plant the peanut or the bean or whatever, the pea into the ground, what will happen is that those cotyledons, they will actually, not actually, that's a bad drawing. What will happen is that it will start to grow, and the two then it will have the normal leaves, and the two cotyledons that once were part of the seed actually are down here. They don't look like normal leaves, but they turn green. They become green, and then you have your little plant here, and then in the middle of here, of course, you have the roots growing, side roots. In the dicot, you always have a main root and then side roots. So that's kind of the idea here. So if you would think of a seed, think of a peanut, because that's easy for you to remember. And I wonder, in the peanut, where is the fruit of the peanut? Where is the fruit of the peanut? What do you think? Well, that's the thing that you remove, okay? From when you break open a peanut and you remove this woody-like thing, that's the fruit, okay? So, the next thing, I'm gonna move on a little bit here. If you want to germinate seeds, basically factors needed for seed germination. If you want to grow your own plants, what do you need? There are three things that you need. Do you need light? No, generally not. Seeds cannot do photosynthesis. They will do, only cell respiration. So you need the right temperature. That's pretty important, because cell respiration takes place. And cell respiration being an enzyme-driven process, you have to have the right temperature. Then the second thing is cell respiration. What do you need for cell respiration? Oxygen gas. Okay, a germinating seed, before it has formed leaves for photosynthesis, it will require more oxygen than it produces. And the last thing, of course, is you need water, because water will trigger a reaction that will actually cause the seed to germinate. So this is pretty important. So light. I know there are some seeds that do need light, so germination starts. But this light is not needed for photosynthesis. It's simply there for germination to start. Okay. So what we have to do now is we're going to also look a little bit at an experiment. Okay. And the question is how do you actually determine this, whether seeds actually need the right temperature, oxygen, or water? And what you do is the following. Of course, you have two containers. I'm just going to make two containers here. Okay. So let's design a little experiment. The Erlenmeyer flasks. It's a pretty bad drawing, but you get the idea. Okay. And what you have in here is you place here some cotton wool, okay, also in here, okay, and down here what you have is it's moist, of course, and here you have water, H2O, and that's cotton. So you have to somehow make sure that this kind of is suspended in here. And you have in here air. And this air has 20% oxygen gas, O2 gas. And what you have over here is the same thing. You also have cotton. And what you have in here is you have not just pure water, but you have a solution that absorbs oxygen. It's an alkaline pyrogallol. Yeah? So this absorbs oxygen. So how much oxygen do we have up here? We've got 0% oxygen. Okay, because yeah, of course you have to have a stopper on here at the top. Yeah, and so the oxygen is removed, and the cotton, which is moist. Why does it have to be moist? Well, remember I told you that it needs water. Okay. Yeah, and then we have the question is, does it need oxygen? Of course, everything has to be at the right temperature. Yeah? And you place over here, you have your seeds here. Yeah, maybe I should change the color a little bit. Yeah, in the cotton, you have your seeds. And then you can basically check it. Everything is controlled. The only difference is that over here you have 0% oxygen, and on the other side you have 20% oxygen. Yeah? And then so this one here should germinate, and this one here should not germinate. That's it. Yeah. So what is the whole point here? The whole point here is a little bit to illustrate or a method of actually removing oxygen. I mean, you can do something similar. If I were to do this, I probably wouldn't use the solution that absorbs oxygen. I would probably flood the whole thing with CO2. Yeah, that would probably be easier, I think. So there are different ways of how you could possibly do that, or pure nitrogen gas or something like that, right? But essentially it shows that you need those three things here, okay? So the next thing is, I'm already out of, am I already out of here? This is what I'm going to do is I'm going to add another foil somehow. So here we are again. I would like to talk about another thing. It's called micropropagation of plants. It's something done in the lab. I mean, normally you grow plants out of seeds, as I already mentioned it, but there are also other ways of how to grow a plant, and sometimes you do this for the following reasons. You know what, I'm going to explain it to you later. I'm first going to show you what is done, and then I'm going to show you why they do that. What you do is you start off with, it's supposed to be a flower pot, you start off with a plant, okay? Okay, it's pretty obvious. Okay, you have a flower pot plant, and then what you do is the following: is you prepare a test tube. So I'm going to prepare a couple of test tubes here. Okay, I'm going to prepare. It's over time. Okay, it's not one test tube. I'm just There's not 3 test tubes, it's actually only 1 test tube that actually shows what happens over time. And what I have in here is I have some kind of a nutrient medium. Okay, and what it's actually, it's sterile, it's actually not even nutrients. It's a gel-like substance called agar. Okay, is in here. And what we have in here as well is a lot of auxin. You already know this hormone. Okay, this plant hormone that causes the plant to grow towards light. And what I do is I take a little bit of this plant tissue and I put it on here. Yeah, so it's a small amount of plant tissue. Okay, and what the auxin will do, it will cause these cells to divide. And so basically plant tissue starts to grow because we added this hormone. And then what will happen is after some time it will start to grow this weird amorphous, doesn't look like a plant, ball of cells. Okay, and we call this a callus. And this callus is it's doesn't really look like the plant, but it starts to grow. It's kind of like a lump of cells, of plant cells. And then what we will see later on is that this callus will actually start to slowly grow roots and leaves. And what we will have to do here is a plant starts to grow. And what we do is we transfer it to a medium with less oxygen, because we want to make sure that the plant starts to grow normally. And sometimes we also add another hormone, a plant hormone called gibberellin. And it's another hormone. Yeah? And as a consequence, so then we replant the whole thing, and what do we have? We again have a plant. And believe it or not, this plant is genetically identical as the first plant. So this is plant A, and then we have here plant B. And plant A and plant B are, of course, clones from each other. They're genetically identical, obviously. Why am I showing this to you? Well, of course, simply to show you the importance of plant hormones, what plant hormones can play a role, and that you're able to actually grow plants also out of plant tissue without seeds. Okay. The question is now, why do they do this in a lab? Okay. So what's the whole point? One of the reasons is it's very efficient. So purpose, maybe I should write purpose. Purpose of micro propagation. Okay. And one of the reasons is you can very fast production of new varieties, plant varieties. I mean, just imagine, maybe you're a company, and then you've just over many years you've made this beautiful plant which is able to produce excellent fruits. Okay, and now you want to sell the whole thing. And how do you do that? Well, the first thing is, okay, I'm going to take the seeds. Nah, you don't do that, because the seeds are not all the same, and then you have to use another plant to fertilize the ovaries, the ovules, and then you have a new plant again, because it's sexual reproduction, right? No, I want to have a genetically identical clone of this, okay? Not by again breeding it. So what can I do? Well, I just do this here in a lab. I just chop up this first plant, and then have, I don't know, hundreds of genetically identical plants. Yeah. Another thing that is also very important is it can be virus-free. Virus-free. There are plant viruses. Okay, and these basically you can produce virus-free plants, and these viruses might reduce plant growth. So it's also good for agricultural purposes, right? And for example, you can also, it's not only fast, but you can also produce large numbers. Apparently they do this for the flowering plants called orchids. Okay, and these orchids, which you can buy relatively cheaply in a shop, basically, they were micro propagated. Yeah. So you do not want to take them from a wild habitat, but you would like to grow them very quickly, efficiently in the lab. I'm almost finished, but I just realized that I actually did forget one short topic, and I probably would quickly mention this as well here. So I have to go back to the flowering, and this is the topic of day length. So this has nothing to do with the micropropagation. It's simply something that I just forgot to talk about. Day length and flowering. There are actually two types of plants. They're called, depending on whether they're called short day plants. And you've also got long day plants. Yeah, and the short day plants, they will only flower when the day is short. Okay, and so during winter, flower during winter, and of course here they flower during summertime. And what they found out is that you have there's a certain critical. What is the important thing here is not the overall length of darkness is important, but you're not allowed to interrupt the darkness. So if there's a short flash of light, then this acts like a reset button. Okay, it's the uninterrupted duration length is important. Yeah. So basically, this means that if you have a short day plant, which flowers when the day is short, then the important thing is that if the night is briefly interrupted, then it will also not flower anymore. It's kind of the you need I don't know. I'm gonna say maybe you need 13 hours of darkness. Okay. So this is a short day plant. 13 hours of darkness is needed. But if you give it, let's say, 7 hours and 6 hours, but there is a short interruption of light between here, then essentially it means that it will not flower anymore. It will think it's going to reset, and it's going to, even if this is very short, then the plant's going to think that there was again a day there. Okay, so the important thing here is not the duration of the light is important, but the overall uninterrupted darkness. Yeah? That is important. So yeah, this was kind of just a short comment, and with that, I'm finished with plant biology. Okay, wish you a nice day. Bye bye.